Bismillahirrahmanirrahim. Today I am going to discuss red blood cell morphology, count and functions. Red blood cell. Red blood cell are the most common type of blood cell and the principal means of delivering oxygen from the lung to the body tissue. Red blood cells are also known as RBCs or erythrocytes from Greek word erythros for red and kytos for hollow, which cyte translated as cell. A schistocyte is a red blood cell undergoing fragmentation or a fragmented part of the red blood cell. Color of erythrocyte. The red color of erythrocyte is due to the heme group of hemoglobin. The blood plasma is straw color alone, but the red blood cell change color due to the state of hemoglobin. When combined with oxygen, the resulting hemoglobin is scarlet. Oxygenated blood is scarlet or dark red. And when oxygen has been released, the resulting deoxyhemoglobin is dark, appearing bluish through the blood vessels. Erythrocytes in mammals are anucleated when mature, meaning that they don't have a cell nucleus and thus have no DNA. In comparison, the erythrocytes of nearly all other vertebrates have nuclei. The only known exception is salamander. Mammalian erythrocytes also lose their other organelles, including their mitochondria, and produce energy by fermentation via glycolysis of glucose followed by lactic acid production. Rolex formation. In large blood vessels, red blood cells sometimes occur as stake. Stake means plate side next to plate side. This is known as Rolex formation. It occurs more often if the level of certain serum protein are elevated, as for instance during inflammation. This is a stake formation. It is just like when we put books in the cupboard or clothes in the cupboard. In this case, the flat surface of the RBC come in close contact with each other. Normally, they repel each other, but when in certain diseases like inflammation, these flat surfaces come next to each other and form this formation, which is called stake formation or Rolex formation. Blood. Normal value in male is 52 lakhs or 5.2 million per cubic milliliter. In normal female, the count is 47 lakh or 4.7 million per cubic milliliter, plus minus 3 lakhs. 40 to 45% of the blood volume, red blood cell or found element is 40 to 45% of the blood volume. 1 drop of the blood is 1 cubic millimeter. Shape of red blood cell. Mammalian erythrocytes are biconcave disc, flattened and depressed in the center. Here you can see that they are depressed in the center. Size of RBC. Red blood cells have a mean diameter of approximately 8 micron. From here to here, this diameter is 8 micron. This is the diameter of RBC, which is 8 micron. Thickness 2 micron at thickest point. This is the thick point, and at this area where no concavity is present, this area is thick 2 micron. Size at thin point 1 micron at thinnest point. The average volume of the red blood cell is 90 to 95 cubic micrometer. This is the thin point or the concave central point, which is 1 micron thin. Count. A sheet of a paper is approximately 75 micron thick. Therefore, 10 erythrocytes could be aligned side by side across the edge of a sheet paper. This is the paper edge, which is 75 micron thick. And when we keep in mind that the size of RBC, we can align 10 RBC along the edge of the paper. This is a picture of RBC under microscope. As the RBC is thick at periphery, it looks dark, and as it is biconcave or thin in the center, so it looks pale. When RBC is observed under microscope, it gives this picture. Quantity of hemoglobin in the red cell. Red blood cells have the ability to concentrate hemoglobin in the cell fluid up to about 34 gram in each 100 milliliter of cell. The concentration does not rise above this value because this is the metabolic limit of the cell hemoglobin forming mechanism. Furthermore, in normal people, the percentage of hemoglobin is almost always near the maximum in each cell. However, when hemoglobin formation is deficient, the percentage of hemoglobin in the cell may fall considerably below this value, and the volume of the red blood cell may also decrease because of diminished hemoglobin to fill the cell. When the hematocrit, the percentage of blood that is cell normally 40 to 45%, and the quantity of hemoglobin in each respective cell are normal. The whole blood of the men contain an average of 15 gram of hemoglobin per 100 milliliter of cell, 34 milligram in 100 ml of packed cell, and 15 gram in 100 ml of whole blood. For women, it contains an average of 14 gram per 100 milliliter. Oxygen carrying capacity. Each gram of pure hemoglobin is capable of combining with 1.3 milliliter of oxygen. Therefore, a normal man, a maximum of about 20 milliliter of oxygen can be carried in combination with hemoglobin in each 100 milliliter of blood, and in a normal woman 19 milliliter of oxygen can be carried. Total surface area of RBC. The total surface area of RBC is about 3800 square meter. This area is 2000 times greater than the total surface. If we spread the RBC on a surface, it will cover 38,000 square meter area, which is 2000 times greater than the body surface area. Characteristic. The shape of RBC can change remarkably as a cell passes through any shape, like a balloon. The balloon, we can change the shape of balloon to any shape. The same is the RBC, which can change its shape to any form. It is because the cell has great excess of cell membrane for the quantity of material inside. Deformity does not rupture the membrane. We press the balloon, it doesn't rupture. In the same way, the RBC also deform and doesn't rupture. Red blood cells in the life are highly deformable and are able to squeeze through minute gaps. A large RBC, the size is large in the size of the capillary, but it changes its shape and passes through this narrow capillary. This is 400 times magnified picture of a vessel in which RBC are moving or circulating and changing its shape. You can see various shape of RBC in this narrow capillary, oval, round, cylindrical, and pyramid shape and elliptic. So when the RBC passes through a narrow capillary, it changes its shape according to the shape of the capillary. Life history of erythrocytes. Under normal condition, 2.5 million erythrocytes are destroyed every second. This amount seems staggering loss of RBCs until it is realized that the loss represents only 0.0001% of the total 25 trillion RBC contained in normal adult circulation. Furthermore, those 2.5 million RBC are replaced by the production of an equal number of RBC every second. Lifespan of RBC. The RBC lacks a nucleus, so it has no power of repair and reproduction. The days are strictly numbered. Every lifespan is 120 days. It has been calculated that each RBC travels 175 miles in the course its approximately short life. Function of RBC. The primary function of red blood cell is to transport oxygen from the lung to all cells of the body. RBC contain protein called hemoglobin that actually carry the oxygen. In the capillary, the oxygen is released to be used by the cell of the body. 97% of oxygen that is carried by the blood from lung is carried by hemoglobin. The other 3% is dissolved in plasma. Hemoglobin allows blood to transport 30 to 100 times more oxygen than could be dissolved in plasma. Hemoglobin combines loosely with oxygen in lung where the oxygen level is high and then easily released in the capillary where the oxygen level is low. Carbon dioxide is formed in the cell as a byproduct of many chemical reactions. It enters the blood in the capillary and is brought back to the lung and released there and then exhaled as we breathe. RBC contain an enzyme called carbonic anhydrase, which help the reaction of carbon dioxide and water to occur 5,000 times faster. Carbonic acid is formed, which then separate into hydrogen ion and bicarbonate ion. This is a chemical reaction. Carbon dioxide combined with water and it forms under the influence of carbonic anhydrase form carbonic acid. This carbonic acid is associated with two hydrogen and bicarbonate ions. The hydrogen ion then combined with hemoglobin and the bicarbonate ion go into the plasma. 70% of the carbon dioxide is removed in this way. 7% of carbon dioxide is dissolved in the plasma. The remaining 23% of the carbon dioxide combined directly with hemoglobin and then is released into the lung. Other function besides transport of hemoglobin. The red blood cell contain a large quantity of carbonic anhydrase, an enzyme that catalyzes the reversible reaction between carbon dioxide and water to form carbonic acid, increasing the rate of this reaction several thousandfold. The rapidity of this reaction makes it possible for the water of the blood to transport enormous quantity of carbon dioxide in the form of bicarbonate ion from the tissue to the lung, where it is reconverted to carbon dioxide and expelled into the atmosphere as a body waste product. The hemoglobin in the cell is an excellent acid-base buffer as it is true for most protein, so that the red blood cells are responsible for most of the acid-base buffering power of the whole blood. 2 MCQ also for this lecture. Please pause the video, read the stem and select the right options. For question 1, if you have selected answer A, option A, this is the right answer. And for MCQ 2, if you have selected D, this is the right option. So thank you. Please subscribe the channel and write down your valuable comment in the comment box for the improvement of the channel.